Hello, Combination. My name is Subi Sadeh. I've spent over a decade in medical device, pharma, and combination product development. Our industry feels complicated sometimes. Drugs, devices, clinical trials, submissions, sterilization, validation, design control, risk management, market access, reimbursement. The list goes on. My goal is mastery, so this podcast is to ask questions I have to people who may have the answers. Each week on the Combinate podcast, I talk to someone about their area to further understand and simplify. Whether you're a pharma person trying to understand the next wave of products, or a device person trying to navigate a pharma system you're unfamiliar with, or a newbie in both areas, I invite you to listen, and together we can simplify by combinating. Hi, everybody, and welcome to another special episode of the Combinate podcast. I'm your host, Subi Sadeh. On this episode, we are graced and honored by Fran DeGrazio. Fran's been on the show a few times before. And in this episode, I wanted to go over USP 382 in a little bit more detail. I know, Fran and you and I talked about stopper manufacturing before, but not as it relates to 382. And I think 382 is the looking at stoppers from a combination product lens, maybe more so than anything else. So maybe talk about what problem was 382 solving and what is the difference now between 381 and 382?
Sure. So typically in the USP for years, it was the USP 381, which was really focused on physical chemical characteristics and really still is. Okay, so that's what USP 381 is. It's more around the chemical characteristics of elastomeric closures for injectables.
Broadly speaking, including a vial stopper or a syringe plunger, doesn't
Yes,
matter.
any
Any rubber.
closures. Yep. 381 came to be, 382 rather, came to be because I think there was a recognition, certainly from the people that were part of the group that were working on this, that it's fine to look at components and you need to in isolation. You need to understand certainly the characteristics of your individual packaging components, but ultimately these things are all used as in a system. And so I know, Subi, you alluded to combination products, but really, you need to think systems, whether it be a stopper and a vials together as a system and a seal, okay, or it's a plunger and a syringe as a system and certainly as a combination product. That's really ultimately why 382 was designed. It was working with this whole thought process now that is around more of a risk-based approach, focusing more about how are you actually going to use this product? What is this packaging component going to be used with what glass? Because that's going to matter in how it functions. And what's the siliconization level of the glass, right? Or how is it all going to be sterile? All those things have an impact on functionality. And so I think they did a really good job of raising that awareness to the industry. One of the challenges that may come out of this, though, is that I think typically, quite often, pharmaceutical sponsors were looking to their suppliers to provide them with the USP 381 data, for instance, and maybe you could do that with the physical chemical data as a standalone. But now you need to generate data on the system. So unless you get your or a supplier or service provider to test that system for you, ultimately the sponsor is responsible to generate that data now to comply to 382.
Are there any I should say this. Broadly speaking, most manufacturers that are producing stoppers are also producing vials, right?
No, not
No, that's not true.
no.
There are some. Yeah, interesting.
Yeah, the stopper manufacturers are really totally separate from the glass manufacturers. Most of them are that way. And that's why even amongst glass suppliers, you have the potential for some dimensional tolerance differences and things of that nature. So you should never just automatically think anything is these two are going to work together, right? You've really got to prove that out.
That's not so I think what I was getting at is more along the lines of, do you see a future where those suppliers that are making both vials and stoppers or syringe barrels and plungers, do you see a situation where they're generating 382 testing in the same way that they generate 381 testing on if they're
Yeah,
making a system, would they also maybe have a body of data for their
their
main
system.
components?
Yeah, that I could see. Yes, definitely. If they have both product types, I absolutely could see them generating the data on both.
Now, one thing that 382 I think speaks to in a little bit more detail is integrity tests.
Yes,
And particularly around sampling. I think CCI sampling was this not so clear. So I don't remember. I think it was like 10, I think on the drug side they were doing N equals 10. Is that right?
could be, could be. Yeah, and I don't remember exactly what's recommended. And again, you have different sponsors that will do different things, of course. But one of the things, and I don't know how true this even is, it's because I am certainly not a statistics person, but we would always say sort of 30 gave you statistical confidence.
20 is what it was.
Yeah,
And this was one of those drug versus device. How can you test 20? What's the edits? Anyways, I do find it interesting that it says select 30 samples per test, which is closer to what you would typically do on the device side from statistical sampling for something like a functional test. But moving on, so there are some elements of 382 that were part of 381 and were removed and
uh,
put into the new.
yes,
Yeah, so like something like fragmentation is something that was there before.
yeah,
So basically, the decision was that 381 is just going to focus on physical, chemical,
yes.
anything related to functional performance where there's an interaction is moving into the new two.
Really took. Yeah, that's correct. And again, I think all for the same reasons. And I know this is challenging, but you talk about pouring. One of the things that can impact pouring the most is the needle being used that's going through that, or how many times, of course, that it's being, the rubber stoppers being punctured. The other thing that can have a tremendous impact on pouring is have the stoppers been processed? Okay. Because remember, rubber is sensitive to heat input, right? Any of that energy input, whether it be through steam or from gamma radiation or anything, you're either building cross-linking of the rubber or you're breaking down the rubber by breaking cross-links of the rubber. And if there is any kind of over processing that's done in on the sterilization side or anything like that, that could have a direct impact on functionality, even of a stop. I know we look at stopper and vial systems as simple systems in comparison to, I think, the more complex combination products, but even those things can be impacted. And I think that's one of the reasons why they want you to evaluate the components as a system, that it's going to be used the way it's going to be marketed. So is this product, for instance, you're using sterilized components and then you're going to terminally sterilize at the end also? That's now a double sterilization cycle. What impact could that have to your components? So these are the kinds of things that they were really thinking about. There isn't a one standard for everything. And I appreciate that very much, because what the regulatory agencies, right, preach now, right, everything that's out there now is about evaluating risk and really understanding the ultimate end application for the patient. And to have one standard for everything, I think is just the opposite of ultimately what should be happening.
Understanding your use case. You talked about the impact of certain processing parameters on a stopper. You mentioned steam and gamma specifically. Do they both make it perform in a way that's worse from a coring fragmentation point of view, or is it?
It really relates to the amount of energy that's put into the elastomer. Typically, we find that a steam autoclave is better than the gamma radiation. However, if it's a controlled lower dose of gamma radiation, there may not be a how controlled is the gamma process? At what level are you gamma irradiating? How many times is it getting dosed? But you can overdose or the energy into the elastomer with either any type of sterilization process.
Um, have you seen, is it already, what's the word I'm looking for? Is 382 already,
I believe
do you know?
it's going to be effective beginning of 2025, if memory serves me right, Subi. I think they were
Or
giving 5 years in essence for people to get on board with this.
let me look it up just so I'll ask the question again.
Yeah, because if it's not right, I don't want to give you wrong
Yeah,
information.
yeah, I know.
And my memory is
I just asked you to do this. You're right, December 1st, 2025. All right, I'll ask the question again. So, Fran, is USP 382 already effective?
no, it is not effective. It will come effective in 2025, I think December, if I remember correctly.
Yeah.
And
I was just gonna say, have you seen companies already start using it?
yes, because again, your products are going to have to meet this standard. So they really gave a 5 year transition period, especially since this was a new section that you're required to meet, right? So this is too. It's not optional. It's going to be a requirement. And from that perspective, I think they're really encouraging people to make this kind of transition now and generate the data or work with your suppliers or work with your service providers to do.
Now, one thing that I do like about it is that it has a lot more detail than I think was in, say, 381 by itself, particularly on the, obviously, that's the reason that they did it. But
Yeah.
with all USPs, there's the 1000
Yes.
sections that offer a lot more guidance.
Yes.
Uh, so-
Actually, I was going to mention if you look at the 1000 series now, I think almost for every more recent P update that has come out, they have, for instance, 1381 section, and it will explain and give a lot more background information. So that's really the informational chapter that aligns with now the chapter that you need to execute on.
So one thing that 1382 does is it links ISO to USP in a way that is, I think, quite meaningful. And it also talks about, in particular, which functional suitability tests may apply for. I think basically what the line that I see them drawing is that 382 is the requirement. USP 1382 is obviously guided. So they say, although not mandated, testing components and/or closure packaging delivery systems according to such standards may prove useful. And then they go through and list for sterile hypodermic syringes, for injection containers, for infusion equipment, for sterile use, sterile single use syringes with or without needle, all the ISO standards that may apply. I think it takes a lot of the guesswork out of it. But do you know why they went through this level of assessment?
What I'm seeing is that there's definitely more trying to get more harmonization. There's a lot of activity that's going on in the industry where we're trying to bring together and develop more global standards. Although, okay, officially, this is not a global standard, but I think when you pull in the content, the context utilizing ISO. That's a way of doing that, right? And so I think I've seen a lot more of that kind of activity ongoing with a lot of the more recent even guidances that have come out from the FDA. I think that the USP doing this is very consistent with that thought process.
So in combination products, there's obviously typically a lot of concern as far as certain functional performance tests. Coring and fragmentation is very common. This 382 talks about break loose, plunger seal integrity, tip cap. The spike related tests, I think are, I don't want to say typically less. In my experience, it's usually IV containers and kits that have some kind of vial access device that have that consideration. But something like a pre-filled syringe or a pen or whatever, typically spike is not. So can you just talk about spikes in general? Because I think you
Yeah.
probably dealt with them a lot.
Yeah, actually, Subi, that's a great question because as you were talking, it's exactly what I was thinking of bringing up really next in the context of this functionality, because quite often, again, people look at vial stopper systems as a simple system. And nowadays, especially, a lot of times vials are part of, for instance, a kit, or will go with a CSTD or will eventually be used with a CSTD in a hospital pharmacy. And one of the biggest functional challenges that we've seen from an industry standpoint is that use of a spike with a vial system. And it not necessarily, I'm not talking about large IV bags or anything like that. Of course, I'm talking about a vial with a medicine, and you're just putting whether it be a vial adapter on it or whether it be a CSTD or any other kind of spike. So that functional understanding needs to be there, right? If you're supplying, and even if you aren't supplying a access spike or vial adapter with your vial, and you should have an understanding of or the most common spikes out in the industry, or will they work with this vial system? What are some of the challenges? Some of the challenges is just getting the spike through the stopper if it's not maybe the most appropriate stopper choice, or pushing stopper push in is very common. And then certainly all these things.
Push
Uh,
in as in the stopper caves in as you're,
exactly, exactly.
doesn't.
And there's all kinds of potential sterility issues with that kind of thing. So this is why I think all these standards now are trying to reinforce this. You really need to understand the system. And when you're talking system, in a lot of cases, you're talking that access to get to that medicament, whether it be using a spike or a vial adapter or whatever. So it is generally that is one of I would say one of the weaker areas in the industry. I think that in a lot of cases, many pharma companies have identified these areas, though, and they've been working on trying to characterize their products and understand it and understand what alternatives there are. But if you're going out of the gate and you're choosing packaging, how is the product going to be delivered ultimately? And that is why it's so important to understand that end patient and the end therapy. How is it going to be used? Because you really need to be considering that as part of your packaging choice.
The 1382 goes into kind of the different, just actually it talks about it in 382. Actually, I'll say it this way. In terms of the scope, like this, the scope is very wide and it includes vials and bottles. Obviously, we talked about those. It also includes syringes, pens, injectors, but it also includes blow fill seal containers, IV bags, and things like that. Can you maybe talk about those as we round out the less common,
Yep.
less covered? And I have a lot of experience with IV bags, but in my experience, the combination product space is a lot less familiar with them.
Yeah. And yeah, if you talk about those kinds of items, functionality still holds. right? And when you're talking about an IV bag, you can be hooking all kinds of various mini spikes and containers and things off that IV bag, IV sets that are going to be used with these things. So those things you really need to be characterizing that to understand how is this going to work ultimately in the hospital or with the patient. And the same thing goes for when we talk about blow, fill, and seal, or some of these alternative or newer packaging methodologies. Again, they need to meet the same kind of standards. Just because you go with a blow, fill, and seal uh, product to inject a patient or to store a medicament and then it gets transferred to the patient, it needs to be able to hold on to a spike if that's the case. Or if you're injecting someone with a blow, fill, and seal type of injection system, the delivery needs to occur just like it would as far as the amount of dose, so the dose accuracy and things of that nature. So just because you're looking at alternative or newer packaging or combination product formats doesn't mean that the criteria of suitability changes, right? Because suitability stays the same for the patient.
Fran, as we move to close, I invite everybody to check out your blog, Frankly Fran, but where can people find you?
On LinkedIn, LinkedIn's the easiest way to get a hold of me, and feel free to reach out with questions or other interests. And I appreciate the time, Susie.
Honor's mine as always, Fran.